And without further ado, it's now my great pleasure to introduce my good colleague Firat Guder, who's a senior lecturer in the department, and he will talk about some recent work that he and his team have been doing on rapid detection of pathogens. Firat, the stage is all yours, and I'm looking forward to your seminar.
Fantastic. Thank you very much for the introduction. And thank you everyone for joining in. Good morning, good afternoon, good evening, and I guess very early in the morning to all of you. So I would like to take this opportunity to really talk about some of the work that we have been doing in the past couple of months to address, as David mentioned, a pressing challenge in our society, which is the COVID pandemic. So, and one of the things that fascinates me about this whole problem, that everybody was surprised, and to me, actually, we should not be surprised at all. So the fact that more and more people are living in the cities in close quarters makes the movement of pathogens between people and across the society very, very simple. So in the 1960s, there were approximately 1 billion people living in the cities, and 2018, there's approximately 4.2 billion people living in the cities. Again, in close quarters, these people are taking the bus together, they go to restaurants, so pathogens really can move across between different hosts very, very quickly. And by 2050, we will have about 9.8 billion people living in the cities. So that's going to be oh, I'm so sorry. There we will have approximately 8 billion people living in the cities. It's an enormous number. So the trend seems to be in this direction and is not going to change. So, and we take the issue with living in the cities, or well, let's not call it an issue because it has advantages too, but the concept of living in the cities, and let's throw in a little bit more firewood. We also allow people to travel very, very freely. Now there are flights going, connecting all parts of the world together. So you can go from one end of the world to the other end in a matter of days, and which would not even be possible in the past in months. So a pathogen can originate in one part of the world and can be transported to another part of the world within 24 hours. And as we saw with the COVID-19 pandemic, some of the symptoms may not even appear in 24 hours. So it's very hard to stop things from moving around when we travel just so much. So there were approximately 4.5 billion passengers traveling across the globe in 2019. And I mean, we don't know what's going to happen post-pandemic, but again, the trend was kind of, upward. So meaning more and more people travel because it just became cheaper and more readily available. But and then there is another thing that, again, is a huge indicator of, and it tells an important story, that whatever is going on today likely is not going to be the last time. So between the 1900 and 2000, there were already several big epidemics, or let's say let's call them pandemics even. Millions of people died, and we tried to basically prevent this from happening, and it kept coming back. But and then it turns out that with the number of people living in the cities increasing, number of people traveling increasing, actually the frequency of epidemics / pandemics also, they are increasing. Between, again, in the last century, there were 4 or 5 really major ones. And just since 2000, we have already had the first SARS outbreak, we had the swine flu, we had MERS, we had Ebola, and now we have the COVID pandemic. So the frequency of outbreaks is increasing, and thankfully for now, the severity has not been as bad as the Spanish flu, for example. But it's just that this was something that was bound to happen. And with MERS, for example, it didn't really happen. It was relatively contained. With the original SARS pandemic, the epidemic, the outbreak, it didn't really happen. That it didn't really spread everywhere. It was contained. But just SARS-CoV-2, which is the causative agent of COVID-19, turned out to have the right characteristic to start this whole global fire. The symptoms didn't appear early on, and etc. So, of course, we are not helpless. Diseases, the frequency, they are increasing, and diseases have been with us for a very, very long time. In fact, part of our genome consists of viral genome that infected us in the past. So we can fight back. So there are really two effective ways to fight infectious diseases. One is to provide treatment or cure using vaccines or drugs. And obviously, like with any disease, ideally we would like to have a vaccine or a drug to treat or cure the disease itself. However, the problem with infectious pathogens is that they may emerge out of nowhere, and we would first need to understand these things, and then start the process of developing a drug or a vaccine to cure or treat the symptoms. And this process is very slow, unfortunately. And partially it's because biology is slow, and which means that when we do the experiments, things will take time. But aside from that, I mean, if we look at the vaccines are given to healthy people, right? So that means that they need to first work, and second, they should not make the healthy people sick. So that means that they need to be tested really, really well, and that just takes a long time. So the other weapon that we have in terms of tackling infectious diseases and outbreaks is diagnostics. So diagnostics can be developed and adopted relatively quickly. Essentially within a matter of weeks after the outbreak started, there were already test kits available to directly detect the pathogen itself by detecting its genetic material. So they are very fast and quite versatile. And diagnostics, and when I say diagnostics here, I generally mean molecular diagnostics, although they are not the only ones. So they are important for the identification of the disease, and also allow contact tracing, isolation, and containment before things kind of go out of hand. Because again, there's a lag between the outbreak starting and the development of vaccines and drugs to treat the symptoms and the diseased individuals. So our best strategy is to detect early, catch early, and then try to contain as much as possible. But also, as we start the society back up, it would be good to know who has immunity, right? And that's where, again, diagnostics play a huge role, because they allow us to first contain, but later on also allow us to, get people back at work, or if they are key workers, obviously it's very important for them to be healthy. But if these are if the individuals are just they just need to work to provide for their families and so on, it's actually important for them to be able to go out and carry out their tasks as much as possible. So in an ideal world, there are some requirements for diagnostic devices. We want the diagnostic devices to be very, very low cost so that we can perform a lot of tests, and they need to be disposable as well, because if we take a sample from an individual, we contaminate the device. So they need to be inexpensive, but also disposable, that we can just throw them away. Ideally, we want them to give an answer quickly, and also portable, because some of the labs, the tests that we run right now, the genetic test that's currently run is called the PCR test, polymerase chain reaction, which allows amplification and detection of the genetic material from the virus. So, however, for example, this is a centralized test, which means that a sample needs to be taken from the individual, and then it needs to be sent to a central laboratory. And ideally, we would not want to do this because that's a process that it just takes time. So ideally, we would like to collect the sample, run it at the spot, and then get the answer. And if the person is positive, then we follow a certain course of action. If the outcome is negative, then we basically follow another course of action. So diagnostics, they need to be sensitive, meaning we want to be able to detect the diseases or the pathogens at a relatively low concentration. They need to be specific. We do not want to detect any virus or any bacteria, so any pathogen. We would like to detect the pathogen that we're interested in. So that means that they need to be both sensitive and specific. Another thing that became very obvious in during this current pandemic was that in a global world, supply chains are very connected. So when the pandemic started in for example, in China, already as early as January, we could not order things out of China, right? So, and if we depend on the manufacturing base infrastructure that's in a single region or a single country, it creates enormous problems in terms of continuing to develop the technology and make it available to different countries. So when back in 2006, I was thinking about new approaches for molecular diagnostics, and I got interested in the whole animal diseases because unlike human outbreaks, animal outbreaks are incredibly common. And I mean, when you look at these pictures, they are kind of like mega cities for humans, right? They're just like, you know, in terms of animal farms, we cram a lot of animals together. They're very close to each other, so they spread diseases very, very quickly. And so I was trying to find problems that actually have very tough constraints, so that if we solve this problem, which means that we can always repurpose the technology that we have developed to solve other problems. So, and one thing that I have come up with was really the diagnostic devices for animal diseases, because we cannot value human life. It's invaluable. But it turns out a sheep costs 60 pounds, right? So that means that our diagnostic test cannot be expensive. Otherwise, it's just not going to be used. So, so my idea was to kind of, let's find a tough enough problem with really tough constraints, and then we develop a solution for this. And because diseases, they tend to have similar characteristics. If it works with this, it will also work with other diseases as well, especially in terms of the genetic detection strategies. So and we started developing diagnostic devices for animal diseases because they need to be inexpensive, they need to be portable, and they need to be operated in remote places in low resource settings. And again, there are frequent outbreaks for animals, for animal diseases, so there is plenty of testing opportunity. And there are many similarities between human disease and animal diseases in terms of the again the transmission and the density in which the individuals in the cities and the animals on farms are kind of live together. And another interesting point for me was that if we develop a device for farms in the developed world, it would actually be a very good model system for the developing world, because a farm in the developed world has roughly the same amount of resources in a city in the developing world. So that's when I realized that this is something we should probably pursue. And back in 2016, we started working on developing a molecular diagnostic device for detecting the genetic material of pathogens in animals that affect animals. So this is what we have come up with. and I understand it's just too many words, but I'm going to describe what I mean. So we have developed a diagnostic device that detects the genetic material of pathogens. And what we really tried to do is we took the conventional method that's used for detecting pathogens, which is the PCR test. And the goal was to make it very inexpensive and very portable, so the test could be run anywhere, so that they're not they don't need to be sent to a central laboratory. So I have a small video and that describes the technology. So we call this technology the TriSilix, which is a silicon-based device for detecting the genetic material of pathogens. So the conventional device is a tabletop device that's 0.5 meter in size. And what we have developed is a tiny device that you can hold it with your finger. So the device itself consists of a reservoir that holds the sample and the reaction reagents. And then, but what's really important is the bit that's at the bottom, which is a silicon-based integrated transducer that has 3 modes of operation and 3 modes of functions integrated into a single device. So it can act as an electrochemical nucleic acid sensor, heater, and a temperature sensor. And all these functions are needed in order to be able to perform PCR type detection. So the top part has the electrochemical sensor. As the DNA is amplified, so we use a certain chemistry that allows us to detect the concentration of DNA as we increase the numbers of copies of the DNA that we're interested in. It produces a signal that's relative to the concentration of the DNA molecules within the sample. So the signal generated is related to the concentration. But in order to do the reaction so that we can make more copies of DNA, we actually need to be able to heat the sample itself. And this is called temperature cycling. So we need to heat it at different temperature for different amounts of time. But we need to maintain the sample at a set temperature. So that means that we also need to be able to determine what the temperature of the sample is. So the device that we have produced also allows us to do this. So you can heat a sample, can detect the temperature of the sample, and it allows us to measure the concentration of DNA that's basically amplified within the sample. So that's kind of what we have developed. Okay, moving forward. So the device that we have developed is a silicon-based device, and silicon, as some of you may already know, it's often it's the material that's used in the semiconductor industry for making chips that go onto your smartphones and computers and so on. And the reason why we chose silicon as the material is that it's a highly processable material, and already there are many methods exist for fabricating it with high precision. That means that we can make very precise devices, very controlled devices using this technology. So in addition, silicon has many intrinsic properties that we can easily exploit. So it's a semiconductor, so that means that we can pass a current through it and easily convert it to a heater, like the heaters that you may use in your homes in the winter for heating. And but aside from that, because it's a semiconductor, its conductivity, its electrical conductivity actually changes, or it's related to temperature. So that means that if we measure the electrical conductivity of silicon, we can determine the temperature of our sample. So it, just as the material itself, without doing anything, it provides 2 of the important functions to be able to build a PCR type DNA detection technology. And the material itself is biodegradable, and it's inexpensive too. A silicon wafer, a large silicon wafer, can be bought for as low as $10. But it has one huge disadvantage, silicon technologies. It's very expensive to process silicon because they're often processed in a clean room, like the person in the middle here. You need to dress up. It needs to be a clean environment. and the machines that are used for processing silicon, they are quite expensive. Of course, it's beyond the scope of this talk, but technologies such as lithography and vacuum etching and deposition processes are used for building silicon-based devices. So can we take all the disadvantages and eliminate them so that we can exploit all the advantages of silicon? And that's what we really tried to do. So, okay, if we want to make low-cost silicon devices that do not need to be processed in a clean room in, let's say, in Taiwan, Japan, or some other developed industry in the US or Europe, we need to come up with a fabrication strategy that could be performed anywhere. So we said, okay, patterning, it's super important to make the devices. Well, can we replace patterning with laser cutting? Because laser cutters have become quite accessible nowadays. Deposition, can we somehow eliminate vacuum-based technologies and instead use solution-based the nanostructuring and then electroplating slash thermal bonding processes that could just be performed in our standard laboratory? And then etching. So we would like to ideally just use, again, wet processes, wet etching to structure the substrate. And then once we make the devices at wafer scale, we don't want to use a diamond saw. Again, we use the same laser cutter that was used for patterning, for dicing and making the individual chips. So here we show on the left-hand side one of the devices that we have produced. So this is the TriSilix chip with the reservoir on the top. As you see, it's a small device that intends to replace the large tabletop devices for PCR detection. And the process is a wafer. It's a batch process. That means that we can use a whole wafer. Whole wafer is just a silicon wafer. And then with a single wafer, we can make up to 37 chips. And this is a 4-inch wafer. We can make the wafers bigger. And then, which brings the cost down to approximately 30 cents per device. And the price could further be driven down by increasing the scale, meaning larger wafers, and making the devices themselves cheaper. So with this technology, again, it's a completely disposable technology. The sample is added, and then the test is run, and then after that, the whole silicon chip is thrown away. And the fabrication, one of the enabling things about fabricating these chips was actually to create a nanoporous silicon surface, which allowed us to electroplate materials on silicon very easily in the lab. Because if you have a silicon wafer that has a very flat surface, it's really hard to electroplate metals on silicon, on a flat silicon surface, because they just peel off. So we have come up with a strategy to overcome this by creating a nanoporous surface so that when the metal is electroplated, it creates interlocking structures. And then aside from that, when we do thermal bonding, again, there is structure on the surface that if we take a thermoplastic film, we can just simply heat press it and then create a barrier layer or an insulation or an isolation layer. So we went from all kinds of vacuum-based deposition processes to something as simple as a wet etching process and wet deposition / thermal bonding process. And this is really the backbone of the TriSilix chip. These 2-3 steps. And the final device itself, as I mentioned, it has 3 modes of operation. And then here I show the components that are needed to operate the device, as well as the device itself. So on the top surface, on the top side of the chip, we have an electrochemical transducer. And then in the middle, we have the silicon chip that acts as the electrical heater and the temperature sensor. Oh, by the way, the electrochemical transducer is nothing but 3 metal electrodes, 2 silver-plated copper electrodes, and 1 gold electrode. And then we have the silicon chip itself, the silicon material itself, as the heater and the temperature sensor. So we basically do nothing. We don't need to add any other functions to implement a temperature sensor or a heater. And then we also, at the bottom, we have 2 electrodes to really, that we can pass a current through the device itself, and at the same time, measure the conductivity of the device to see where it lies in terms of its temperature. So, and we need some electronics, obviously, to operate the device. So to operate the electrochemical cell, we have a 3-electrode potential. We have a potentiostat, and then for to heat up the device and to sense the temperature of the device, we have some electronics that are connected to the bottom 2 electrodes. So if we look at the bottom section here, to drive the electronics, we have actually, so to drive the chip for heating and also for temperature sensing, we have developed some custom electronics in my lab, and which allows us to drive an electrical current through the device in a controlled manner, but at the same time allows us to precisely determine the resistance of the device. And if we want to see how well it works, we basically just use the thermal camera to set the device, set the temperature of the device, and then maintain the device at a set temperature. So we can essentially modulate the current that's passed through the device to maintain the system at a certain temperature. And in terms of temperature sensing, within the range that we're interested in, approximately between 20 degrees and 100 degrees, the devices produce a linear temperature dependency in terms of temperature sensing, which is quite useful because nonlinear relationships are a little bit more difficult to handle. And we can cycle the temperature and then maintain it at a set temperature very easily. And we have not optimized, but this, as you see, the ramps are quite quick, which means that we can actually reach to a set temperature very quickly and then do the reaction quickly too. We have also designed a holder that would house the chip itself, and that would allow us to drive the reaction and then detect the genetic material from the pathogen. And on the top part, again, we have the 3-electrode electrochemical cell that allows us to detect the DNA of the pathogen or DNA related to the pathogen. So we did electrochemical characterization of the device. So the way it kind of works, electrochemical detection of DNA, is that we have molecules that interact with the DNA. So if we have these molecules in the solution, their concentration, the concentration of the free molecules that actually could participate in a reaction with the surface, they change with the number or with the concentration of DNA that's in the solution. So that means that if we make more copies of DNA, that means that we will have less of the signal. So, and we have done some characterization to show that our electrodes are indeed, they work really well, and they're sensitive for detecting low amounts of the reporter molecule, which is the molecule that, again, interacts with the DNA, which would allow us to detect low amounts of DNA in solution. So another interesting thing is that because we have a heater, which means that we can change the temperature at which we perform also the detection itself. And it turns out that as we increase the temperature of the device, actually our device becomes more sensitive because the species, the reporter that we detect, can diffuse onto the electrodes a lot faster. So and with this configuration, we also have the flexibility to detect low concentrations of the reporter, which is called methylene blue, down to small micrograms per milliliter concentration. So so we can really detect very small amounts of the reporter itself, which means that we can detect very small amounts of DNA. So and if we put basically this whole thing together, we can start doing microbial pathogen detection. So the first target that we chose, this was our initial target. This was not related to COVID because it did not exist back then, was to develop a diagnostic device for Johne's disease. It's a disease that affects livestock, mainly cows, actually. There is no cure. And once an animal is infected, it dies. And or as soon as the farmer basically diagnoses the animal with this pathogen, usually the animal is culled, so the animal is killed. And there is an ongoing, actually, epidemic in Europe for Johne's disease. Milk producers are worried about it, and then beef producers are worried about it, etc., etc. But it turns out this pathogen is a potential zoonose, meaning it can go from animals to humans. Although it's not fully confirmed, but there is some compelling evidence that there is association between Crohn's disease, which is an inflammatory bowel disease, and the pathogen, which is the Mycobacterium avium subspecies paratuberculosis. Sorry about the long name, which we call just MAP. So there seems to be a relationship between also Crohn's disease in humans and MAP. So MAP causes Johne's disease in animals, but it turns out actually it may also be causing Crohn's disease in humans. So and that's one of the exciting things about the diagnostic technology, because again, once you develop it for animals, it may either be directly applicable to human diseases, or with small variations, it could be adapted for detecting animal diseases. And even we saw with the COVID pandemic, the diseases, again, the SARS-CoV-2, the causative agent of COVID-19, it actually originated in animals and it passed on. So it is actually quite useful to be able to detect diseases in animals. So without any kind of amplification, we were already able to detect approximately 0.8 picograms of 800 femtograms of DNA in solution. So this is genomic DNA of this is a bacteria, by the way. This is the genomic DNA of the bacteria. So without doing any kind of amplification, we could already detect very small amounts of the genetic material from this pathogen using our chip, which shows that our chip is quite high performance. And then we can do amplification, which means we can actually increase the number of copies and make it even more sensitive of a test. So within approximately 20 cycles of amplification of DNA, we could already go down to approximately 20 femtograms of DNA in solution. And that's a very low amount. That indeed is a very low amount. And in fact, it performed similar, but actually even better than the conventional test that's run with the desktop style laboratory device that could basically run the same test. So and in terms of to make it, I guess, more relevant, 20 femtograms, well, what does that even mean? 20 femtograms is approximately, for this pathogen, it's approximately equivalent to 1 microbe. So in our solution of 5 microliters, we can detect a single microbe, essentially, with our technology. So when our MAP project was coming to an end, the SARS-CoV-2 emerged. The COVID-19 pandemic emerged first, obviously, in China, and then spread. And we're like, okay, the initial premise was that these technologies should be readily adoptable. Let's check that. And in fact, the pathogen itself was already sequenced at that stage, and the primers, so the targets, let's say, they were kind of developed, and we could really just read this from the literature and order the proper reagents and run the test ourselves. So here we detected these are synthetic samples. We did not work with the real samples, but we're able to detect the complementary DNA, not the RNA, but the complementary DNA from the SARS-CoV-2 virus. So this is still work in progress, but it already shows that it's highly promising to be able to detect. So, but not only we can detect the genetic material from SARS-CoV-2, the pathogen that causes COVID-19, but we can also tell the difference between the pathogen from the original, the first SARS outbreak, and the recent one. So not only we are sensitive, we're also quite specific to a certain pathogen. And of course, there are many other methods. Maybe this is not so relevant, but there is PCR, which is a cyclic amplification and detection technology or approach. But there are also isothermal, meaning you don't really need to move the temperature up and down. You just keep it at a set temperature. And one of these is called RPA. So our technology not only is compatible with PCR, which is a more difficult technology in terms of implementation, but it can obviously also implement RPA, which is just another amplification and detection technology. Okay, so the future is, again, this is work in progress. Our final aim is to have a handheld point of need device for detecting pathogens directly in the field. So we don't want samples taken from people and then sent to a lab, because clearly people are just going to go, they're not going to stand and then wait for your result to come back at the same spot for a couple of days. So we would like to eliminate this whole process and then do the testing directly at the spot, which means that because it requires no infrastructure and very basic operation, it could be implemented and used in poorer regions of the world or developing regions of the world. So it's quite important to be able to develop a very portable system. We still need to do on-chip lysis. For those of you that do not know what lysis means, essentially once we have the virus or whatever the pathogen may be, we still need to break it open to get the genetic material out. And then we would like to be able to do all this on our device, but we have not implemented this function yet. So that's still some work that needs to be done. We still need to complete our handheld analyzer and then try other electrochemical reporters. We tried methylene blue in this one, but it has some stability issues. So we might be able to, there are other alternatives that we would like to try out in the future. So, and of course, there is one crucial step for RNA-based organisms, or RNA viruses, I guess, that we still need to convert RNA to DNA on the chip itself to be able to, again, make it useful for detecting RNA viruses in the field. And this is another function that we have not implemented yet. So I would like to thank my funders, and I'm sure you have questions. You're like, what was PCR again? Just come and ask, please. I fully understand that some of you are just high school students or just graduating, and please do feel free to ask questions. There are no bad questions. And so all this work was obviously it was made possible through generous funding of various sponsors. And this is my lab that basically do all the work. I just get credit, right? So these are all the people that are actually building these things in the lab. So I would like to thank them. Estefania was the lead author of this work. She built the devices. Tarek helped with the animation that you saw. And then obviously I have fantastic collaborators that we as bioengineers, we work very closely with other disciplines and experts from other disciplines. Karen here is an expert in MAP, the disease that causes is the pathogen that causes Johne's disease. And it's very actually interesting to work with other experts because it's so easy to really just learn from them if you work closely. And there are some things that it's just really hard to learn from books. So you really need to work with the experts in the field to learn how some of the tricks are done, or what the tricks are actually. So all this work was published in a preprint journal and is currently in peer review. So if you want to learn more about it, feel free to check it out. If you are interested in sensors, and my lab mainly develops sensors and actuators, and we have recently published a paper. Again, it's intended for a broad audience. It's called Disposable Sensors in Diagnostics, Food and Environmental Monitoring. It really covers all the basics of the field of sensors in applications for application diagnostics, food and environmental monitoring. So please do feel free to read it. I'm sure even if you don't have much knowledge about this field, you will learn quite a bit by just reading the introduction and the conclusions. And of course, this was just a small part of what we do in my lab, let alone it's just a small part of what's being done in the Department of Bioengineering. We do a lot of amazing and diverse work that are very highly collaborative. So please do feel free to reach out and check it out. And I think that's it. I took actually longer than I thought I would. So I'm just happy to answer your questions. Thank you for listening.
Well, thanks very much for that on behalf of the audience for this very interesting and engaging talk. I'm sure there are many, many questions. So please either, well, raise your hand or ask your question in the chat and I will put it to Firat now. So while people motivate themselves to raise their hand or type the first question for it, maybe I can start. One thing I particularly enjoyed was the mix of very fundamental science somewhere and then actual engineering challenges, right? So you sort of have to really address both. And you did cover it in your talk, but maybe for the audience, it's not necessarily always that easy to realize where they sit. So can you maybe quickly summarize what the key scientific challenge and progress is in that work, and then what the key engineering challenge is, because that's what makes this really unique, right? That you have to bring these two together somehow. Yeah,
well, I think the scientific challenge is really to address address the need for running tests very, very quickly, so that we can actually, can we understand because right now testing takes a really long time, which means that we don't really understand even like how pathogens spread across the society. So if we can provide such capabilities, we would actually be able to not only provide the means of knowing whether someone is positive or not, but we would be able to learn more about how various diseases spread and how they evolve during the spreading phase. So and in terms of the engineering challenge, I mean, there were just plenty. So just taking a process that was fully optimized for in a clean room for clean room processing, and then develop alternative processes so that we can produce these chips in a very standard lab. This was an engineering challenge by itself. But on that, and then not only that you produce the devices outside, but they also need to work well. And then it's another engineering challenge. So we cannot just throw a dead fish on the floor and call it cooked. So it has to work, right? It's just basically one problem after the other problem after the other problem that we have to work on, and we have to work with, obviously, with different teams, learn from different people. Everybody has a slightly different angle at solving the problems. Look at them all, choose the best ones, try them out. They might fail, go to the next one, and so on. So there are really many challenges in terms of engineering. But the key scientific findings would really be that if these devices are out there, it would be really useful to have them so that we can generate a ton of data and really understand these diseases better. Thanks, Firat. I have a question from a member of the audience, Timothy Dufour, and the question is artificial in lab RNA to DNA transcription transduction attainable, and how does it or could it help in pathogen detection? That's the question. The question. It's also in the chat if you want to reread it, but yeah, that's a question. All right. Okay. So artificial in lab RNA to DNA transcription attainable. Well, I think by artificial, if the person means using not using enzymes and using alternative materials, as far as I know, no. But just because something is not there, it does not mean that it will not be there. So these technologies may become available in the future, but to the best of my knowledge, it's not attainable at this stage. But it certainly is something that may emerge in the future. And then maybe the second part of the question, Firat, if I just quickly say how, if it would be possible, how would it help or could it help in detection of pathogens? It's the second part of the question. Yeah, so it would definitely be useful because enzymes are always problematic. They have stability problems, and then they need to be obviously produced using most of the time organisms. So and organisms are tricky, and it's just the whole process is it's slow and generally expensive. So if we can come up with, let's say, synthetic alternatives, perhaps inorganic alternatives that could just be produced in batches that are stable, let's say in relative terms indefinitely, of course, it would make a massive difference in terms of the detection technologies. And again, like one of the challenges is if we want to do things in the developing world, there is no cold chain. I mean, one of my colleagues, for example, he developed a device, he developed a test, a sickle cell test, and to be tested in Africa. And when the samples were sent, it sat in the tarmac for a day in Kenya in the summer. Everything basically degraded and became useless essentially. So if we have alternatives to the biologicals that we use in our devices that are more stable and cheaper, it would make of course, it would have a massive impact, especially in parts of the world where logistics and storage is not really well controlled.
Thanks. I encourage again students or other guests to ask questions. Oh, there's one. The question is, Firat, how long does it take for the device to amplify the DNA signals to a confident level to be test positive? So how many cycles does it take? That's the question.
Yeah, so with our device, approximately 20 cycles. And each cycle lasts about 90 seconds at this stage. We have not optimized it yet. So through optimization, we think that we might be able to go down below maybe something in the range of like 60 seconds or even lower. So that's still work to be done. But our RPA, so isothermal amplification methods, the ones that we tested in the lab, they seem to be a lot quicker. So should be able to produce a response well within 10 minutes and probably even 5 minutes. But the issue is that isothermal methods, so the reason why I like PCR, of course, it's a technology that requires more engineering, but all the patents have expired. So the reagents are very cheap, right? So if we are truly interested in making a low-cost device, we don't want to use some proprietary chemistry or reagents. We would like to use the readily available and the most inexpensive technologies out there. That's why I kind of like PCR. And if you think about it, does it really make a difference if the result is given in 5 minutes, 10 minutes, or 20 minutes? I think in terms of the clinical decision making, probably not. So and that's kind of my views on the topic.
Thanks. There's another question by Kai Firat. Do you use the standard reagents used by desktop qPCR machines, or is there any chance of modifying them to optimize them for your machine? And no, if so, yeah. Okay, sorry, go ahead. Yeah.
So yeah, no, I mean we use the standard reagents because again, we want to create a device that's fully compatible with what everyone is already doing, because it turns out actually probably creating technologies is much easier than changing people's habits, right? And gaining trust. So and that's one thing that's often ignored in engineering and physical sciences. You may have a technology that's just incredible, it's cheaper, and then it works well, and etc. But if people don't want to use it, they just will not use it. And so what ideally we would like to do is, look, this is everything that you're used to, but it's cheaper and faster and portable, etc. That's kind of it's been my philosophy for developing this technology. And so we have just used the standard reagents.
Yeah, it makes sense to me, at least. So you convinced me with that argument. There's two questions on the methylene blue. One is sort of whether you can generally briefly explain what the role of the methylene blue is, and the other one is whether the methylene blue is conjugated to a specific pathogen sequence. So both of these related to the role of the methylene.
Sure. So I mean, methylene blue is just it's a redox. It's called a redox reporter. So what it really does is that it interacts with DNA, so okay, let's take a step back. What's a redox reporter? So essentially what it does is that it can be reduced or oxidized when a certain potential is applied to an electrode. So that means that it can accommodate electron transfer, so charge transfer to an electrode. So which means that if it is around, if it is on the electrode, what it does is that it allows generation of an electrochemical current. But if it's not around, if it's tied up inside the DNA molecule, that means that it's no longer around to be able to generate a current. So that really is the role of the methylene blue. And the second part of the question was, does it bind to a specific segment? No, because the specificity in the test comes from the PCR process, not the redox reporter. So the redox reporter just says, okay, let me tell you how much DNA is in the sample. That's my job. It doesn't tell you what that DNA is. That part of the detection process, it's really handled by the biochemical amplification of the molecule itself, the DNA molecule. So if, for example, we do not have the target molecule, we will not be able to amplify it, right? Which means that our redox reporter will not be able to interact with higher numbers or higher copies of DNA, which means that the numbers of the available concentration or available amount of methylene blue in the solution will not change because we didn't detect the target. I hope I did not make it more confusing.
Thanks, Red. And then there's two questions surrounding the sort of number of pathogens that you can test and how difficult it is to adapt it to different pathogens, what pathogens you can already detect, how many you've tested, what your success was, sort of around that topic.
Yeah, sure. So the beauty of PCR is that in terms of genetic detection, it really is the first technology that's usually the first test that's developed. So as soon as a sequence is generated, oftentimes people will produce the reagents to target a specific region in a pathogen. So and because we use the same standard approach for the lab-based PCR, ideally it should just work equally well with whatever that could be detected with the lab-based system. So there should not be any difference. And again, our initial results at least indicate that we should be able to detect at the same level or even perhaps better than what the conventional PCR technologies in the lab that are used in labs that could produce.
Thanks, Ray. And then maybe one last question, because we're sort of already at the end of our time. Someone asked specifically about the future, and you addressed this in one of your last slides, of course. You said what the next steps are, but if you laid out the broad future where this sensor will be or this technology will be in 10 years, that might be a good ending point for your seminar.
Yeah. I mean, ideally, we want this technology, further developed and used in the field. So, however, before we can get to the for example, there already. So my lab is quite entrepreneurial. Like we do try to do translation and take the technologies out, but we still have to figure out, some of the critical steps around. For example, we do not want somebody using another device to prepare the samples and then add to this device. We would like to be able to do entire sample prep inside this device as well. For example, if we look at the COVID case, a nasopharyngeal, usually swab, is used to collect the sample from let's say the nasal cavity, to make it simple. So ideally, what we would like is that once the sample is taken, it's ideally put directly into the device and nothing else is needed. So all these things still need to be implemented. And in 10 years' time, of course, it would be amazing to be able to see this technology being used in the field. I'm especially very passionate about applications in the developing world because there are very urgent needs there as well. I mean, the COVID pandemic is just one thing, but there are many other diseases that kill a lot of people, and there are many diseases that kill the animals of people, which is almost a death sentence. It's a death sentence to the people in the developing world, and being that given that animals are more resilient to climate shocks and so on, I mean, this is one thing that they will continue to do. And having a technology that's inexpensive enough that could be used for both humans and animals would really be kind of a dream come true myself, and I'm sure for my team as well.
Thanks. Well, thanks very much, Firat, for this very interesting talk and great Q&A. We are at the end of this seminar, so thanks for all of those who joined. Kemi, is our next seminar next week or in 2 weeks' time? Help me quickly. I don't have the date. I couldn't find it quickly, and I don't know it off the top of my head. Kemi?
You're on mute, Kemi. Kemi, you're on mute.
Oh, apologies. The next seminar is on the 17th of July.
So that is next week.
That's right.
Okay, so next week. So join us again next week, and have a good rest of your day. Thanks again to Firat.
And I would like to mention that if you leave the seminar more confused, then just shoot me an email. I'm happy to catch up and tell you more about what's going on.
No, you might.
Thank you for having me.
Well, it was a pleasure, Firat. Thanks very much, everyone.